Hi there, good afternoon. Welcome to the Dean's Distinguished Lecture this afternoon featuring Craig Cameron, who's a class of 93 in biochemistry, where he did his degree with Jonathan Leese. And I'm delighted that he's here and is co-chairing our reunion weekend with Rebecca Barkus. I'm Stan Gerson for the alums in the audience who may not frequent with me, although most of you get a chance to meet me both here and across the country. And I'm delighted that we get to inaugurate our weekend with this distinguished lecture. I'm delighted that Craig both took on the opportunity to lead us with Reunion Weekend and to provide us with his insight today in his lecture. He's chair of the Department of Microbiology and Immunology at UNC Chapel Hill. I'm going to let Dr. Deal, chair of biochemistry, give a little more introduction on him. Just a little more background about this event. We've had the Distinguished Lecture Series now for a little over a decade. It's a great way to start our reunion activities, get a little academic program before we socialize. And it just reminds us how many of our alums are remarkably successful in their careers. And for those a little younger going through it who will graduate from this institution, we'd like all you to become department chairs, so just keep at it. So without further ado, I'll turn over to Dr. Deal, who is the chair of our Department of Biochemistry and is also Deputy Director for the Case Comprehensive Cancer Center. Dr. Deal.
All right, thanks very much. Yeah, I'm Alan Deal. It's a real pleasure to introduce Dr. Cameron today, a former alum here did his PhD in the Department of Biochemistry. Stan introduced some of his background. I want to go a little bit more into what he's done since being a PhD here, because it's really remarkable. He did his postdoctoral work with Stephen Benovich at Penn State, and for a very short postdoctoral stint, and then moved on to an assistant professor and all the way to the Paul Berg Professor of Biochemistry in 2005, Penn State. You'll notice I'm actually reading this. I never do this, but his CV is so impressive that I really felt the need to use some notes here. Craig is a virologist. He's done just remarkable work. He's published well over 200 papers. His funding is too numerous to mention. His lectures and seminars exceed my wildest imagination. He's been on just more advisory committees than I can count. He's included various HHMI committees, Public Affairs Committee, ASBMB on both minority affairs. It's just remarkable. It's really grateful to see the success of alumni from this remarkable institution, particularly from a department that I hold near and dear to my heart. Craig, really delighted to have you here today and looking forward to your seminar.
Thank you.
Welcome.
Thank you all. I hope I can live up to the expectations of the lecture today. I can say, let me switch this to laptop and go back. Oops. Let's see here. The slides are... Okay. So it's a pleasure to be here. I would not be the scientist I am today if it had not been for the Department of Biochemistry and all of the amazing faculty and my amazing peers. I'd like to start off with this slide because it has really been the people as much as the projects that have kept me excited about science and doing science over the past almost 30 years now. My instincts for science were honed here in the Department of Biochemistry. I was always told that, a sign of a good research program is one that incites more questions than it answers. and that if I was going to really make an impact, that I was going to have to follow my nose and retool as often as necessary to answer the pressing question of the day. And so that's what I hope to illustrate to you and all of the students that might be in the audience today. I left here a protein biochemist slash virologist. I wanted to be a hard, core card-carrying mechanistic enzymologist, so I went to work with Steve Benkovic at Penn State. Then after working on enzymes like dihydrofolate reductase and doing a little work with HIV reverse transcriptase, I started my own lab and decided to focus on enteroviruses, where I had to again begin to think about virus biology, viral pathogenesis, and really the subject of today's talk is single-cell virology and how we've used it to understand better mechanisms of virus replication and pathogenesis. But as you'll see at the end, we're now at a place where I'm gonna have to get deeper into cell biology. I'm 60 years old, and I don't know if I really want to retool again. So let's see. So what I'm going to tell you today in the first part of the talk is my motivation for studying virus infection dynamics on the single-cell level. Some of the puzzles that, existed as a result of doing bulk studies that were illuminated in our initial studies of viral infection dynamics on the single cell level. Where we want to take this, and just some experiments that show that we will likely be able to take this to a stage where we can elucidate between individual variation, the genetic basis for between individual variation and outcomes of infection, without necessarily having to use animal models. And then in the last part of the talk, I'm gonna show you how we're using analysis of cell pairs to understand mechanisms of transmission from one cell to the next. So when my lab started, pandemic preparedness was not a thing at the time, but we were, talking a lot about strategies, broad spectrum strategies to address viral infections. And vaccines were a thing back then, and live attenuated vaccines, how they were developed, was not necessarily rational. A lot of serial passaging and non-human cells are at, non-permissive temperatures. And so what I thought we might be able to do was to apply more logic to development of drugs and vaccine candidates. So the thought was, what if you take a conserved enzyme, very highly conserved enzyme across a virus family, for example, or genus. If you understand that conserved enzyme, the substrate specificity in ways that you can create analogs that would inhibit all of those, you might have good broad spectrum mechanisms. And if you could understand how the chemistry is tuned into the active site of the enzyme, you might be able to manipulate it in ways that would allow virus to grow so that you can make a vaccine, but where that virus would be attenuated. So these were the goals of the lab initially. We decided to focus on the viral RNA-dependent RNA polymerase. Back when I was starting my program in '97, reverse transcriptase was a well-known, very tractable antiviral target, and there was little known about the mechanisms of RNA-dependent RNA polymerases. So we knew that there was going to be a lot of fundamental knowledge that we would glean, even if we never reached our goal of targeting the enzyme. So we chose poliovirus as our model system. So I consider myself a fundamental biologist. I consider poliovirus a model organism like Cerevisiae. And at the time, anything that you wanted to do with a virus, you could do with polio. The community was large, all the reagents were available, decades of biochemistry, facile reverse genetics, and you could study pathogenesis in mouse models. Yes, this is a model system for us. We're all vaccinated against polio, but polio in the genus Enterovirus has a lot of close cousins that still cause problems, and the problems are intensifying. For example, EVD68, Enterovirus A71, these are all respiratory pathogens that are enteroviruses just like polio, and they cause periodic outbreaks around the globe. So one of the first questions that we wanted to address when we started doing biochemistry with the polio system was this question of how important is the viral quasi-species. So RNA viruses exist as a genetically distinct population of variants. We now call them mutant swarms, but back in those days we called them quasi-species, and it wasn't clear on the origin. Was it mistakes made by the polymerase, or were there enzymes in the cell that could act on RNA and change the bases? And today we know that that is possible, right? APOBEX and things like that. But what was becoming clear was that, the mutant swarm is the reason that you have antibody resistance, right, escape. It's the reason that you have drug resistance. So again, being able to intervene if this was a virus-driven phenomenon might represent an important antiviral strategy. So we were able to show quite quickly that the RDRP alone was sufficient to explain the ontogeny of this mutant swarm. We could isolate variants that were higher fidelity or lower fidelity, and that corresponded directly to changes in the population diversity. But the way that we originally made this observation was in our studies of ribavirin. So ribavirin is a carboxamide triazole nucleoside. It was the very first broad-spectrum antiviral nucleoside, and no one really had an idea how it worked. The thought at the time that I started was that it was targeting a cellular enzyme and changing GTP pools in cells, which might have an impact clearly on RNA synthesis, but the thought that it was having an impact on translation, and that misregulating or slowing down translation might create negative interference as the virus was trying to replicate. We were able to show that treatment of cells with the nucleoside converts the nucleoside ultimately to the triphosphate, and once utilized by the RDRP, its incorporation is templated ambiguously by uridine and cytidine, because this carboxamide moiety can rotate. And in one orientation, it base pairs with cytidine, and in another orientation, it base pairs with uridine. And so this gave us a chemical tool to be able to ask just how sensitive is the virus to mutation. And fractional changes in the mutation frequency showed exorbitant changes in, or reductions in viral replication efficiency. So clearly tuning fidelity at this stage could be a mechanism to attenuate viruses. So we went on a pursuit of isolating virus variants that were either anti-mutator, high-fidelity variants, or mutator, low-fidelity variants. H273R shown here. Maybe I should use this thing here. H273R shown here is a mutator virus. And I showed you with ribavirin, I said fractional changes, increases in mutation frequency killed the virus. But here I'm doing a one-step growth curve, standard experiment in virology, comparing the wild type to the mutator. I see no effect. And this is one reason why it's important to be in a system where you can do all kinds of experiments. So we had the model, the poliovirus model, animal model, very permissive model. And you can see that if we characterize the mutator virus in the animal, there was 2 logs attenuation. Furthermore, I had an undergraduate student at one point. We were just doing some evolution experiments in vitro, and what we could see was that if we serially passage polio, wild-type polio, at low multiplicity, it would adapt to growth at high, to high yields from low multiplicity. So the fecundity, apparent fecundity, was increasing. If you used an anti-mutator, a high-fidelity virus, that evolution didn't happen. But if you now use a low-fidelity virus, you see the virus evolve itself into extinction. So under conditions where you have single viruses infecting single cells, you see the impacts of genetic heterogeneity. Some of the mutations clearly are going to be nonsense mutations or lethal mutations. And those are going to not be able to replicate. So at the time that we were doing this, what I've told you here is a summary, is that we have a population of viruses that are heterogeneously distributed, distinct genetically. You also have a population of cells that are being infected, and even HeLa cells in a tissue culture dish have stochastic gene expression when you're thinking about pattern recognition receptors and other proviral and antiviral factors. The way that we do experiments, we do high multiplicity so we can get this one-step growth curve. So the multiplicity of infection is going to be driven by the Poisson distribution. And then each cell is going to have a virus yield. But we're just measuring, the virus that we're putting on and asking how much we pull off. Clearly, what I'm suggesting is that if we had the ability to look at individual cells being infected by individual populations of viruses, we would see distinct phenotypes. So at that time that we began asking questions like this, the tools did not exist to look at viral infection dynamics on the single cell level. And so we partnered with Tony Wong at Penn State, who was a lab on a chip type of engineer, to fill that gap. So what we decided to do was to introduce fluorescent reporters into our viruses. We would make recombinant polio. We would do off-chip infections of cell lines like HeLa. And then we would introduce the cell population into this device. And to get single cell resolution at that time, we diluted the culture. So we had a low enough cell density so that, and a lot of wells, so that we could get occupancy of a sufficient number of wells with single cells that would allow us to do good statistics. And so this is the chip. And we expected, because polio at the time was known to be a lytic virus, we expected that we would see an evolution of green fluorescence as a function of time, post-infection, and that at some point when lysis happened, that the fluorescent reporter, in this case we're showing green, would dilute in the well, and so the fluorescence would decrease. Several years later, we became much more efficient at this. A postdoc in my lab, Wu Lu, was able to introduce traps that would allow us to get 90% occupancy of the wells with single cells. So that really gave us a lot of power. I like showing this slide because what we're doing is not anything that you can't. Oops, sorry. Let's see here. I want to start the movie. So this is just a standard live cell imaging apparatus with the microfluidic chip installed in the jacket. So you can have constant humidity, temperature, and all of this. And we can take time points every 30 minutes for up to 2 days. So the very first experiment that we did showed really clearly the between cell heterogeneity in the infection dynamics. But very interestingly, when we just took the unadulterated average of this population of curves, we actually saw a time course that looks just like we would expect from the population level. So all of this heterogeneity is buried under the bulk curve. So does it matter? That was now the big question. We don't do an experiment like this, developing all the technology, showing you the heterogeneity, and then taking an average. How do we actually analyze the data in a way that we can get meaningful insight extracted? So for this, we collaborated with Klaus Wilke at UT Austin. And every curve has this general type of shape, right? You have a threshold for observing the fluorescence. That fluorescence increases to some maximum. And then, like I said, in certain cases where you get lysis, you see loss of the fluorescence. So what we decided to do was to attribute each portion of the curve to a phenomenological parameter. For example, maximum, oops, let's turn this back on. Maximum fluorescence correlates to virus yield, for example. The slope here correlates to the rate of genome replication. The ability to observe fluorescence, that's like the eclipse phase of the virus replication cycle that we would measure from one-step growth curves. And so what we would do is to measure each parameter in each cell and report those as distributions. And then you would compare the distribution under condition 1, the wild type condition, to some condition 2, which is some perturbation, whether it be a mutation in the virus, a mutation in the host, the addition of a drug, and what have you. So we could do very robust statistics on these distributions. So now let me show you just a few things that were really surprising. So with poliovirus, like I said, it's one of the oldest viruses. It's in every textbook, especially virology textbook. The eclipse phase, well known to be 4 hours. The generation time, well known to be 10 hours. So 4 plus 6 is 10. You get to that plateau. But what we were able to show when we actually evaluated the distribution of the start times was that indeed, at 4 hours, most infections had not been established. Infections were still being established at 5 hours, 6 hours, and some even at 10, where the population average had maxed out. So, we were underestimating the time it took to establish infection. And then when you look at what we're calling the infection time, the time it takes to go from observing fluorescence to the plateau, equivalent to the generation time that we talk about in virology, 6 hours here. But what we see here is on average, it's about 2.5 hours. So the rate limiting event is establishment of infection, but once the infection is established, replication proceeds pretty quickly. So then the question was, what's governing the heterogeneity that we're seeing? Because clearly we're seeing a lot. So we devised an experiment where we reasoned that if we take 2 reporter viruses expressing 2 different colors, and they get into the same cell, and their infection dynamics are highly correlated, for a particular parameter, then it's likely that there's a host factor, the cell, that is constraining the emergence of the fluorescence. But if it's viral genetics, and I'm telling you we have a quasi-species, a mutant swarm, you would imagine that there wouldn't be a correlation between the red and the green. So what did we see? So the first observation, by evaluating the start point, was that we saw a very strong correlation. So for an infection to be established, you have the virus, right, battling the host and the host battling the virus. And if the virus wins, you're going to see replication. And that's why there's this strong correlation. But so the host is driving, host determinants are driving the efficiency of infection, establishing infection, which makes sense. And so many of us have done knockdowns and tried to analyze those experiments, knockdowns of cellular proteins and impacts on not only viruses, but whatever organism you might study. And you only see twofold effects. And a lot of that derives from the fact that you're not actually measuring that establishment of infection. It's being blurred by other aspects. So you can see once polio establishes infection, the yield, the rate, the time from beginning to end, these are all driven by viral genetics, and they're not highly correlated. So it was at this point that we really realized we had something that could be useful. Now let's try to see what types of experiments and differences we can glean from using the single cell technology. And so that takes me to this next part of the talk. So I've been interested in antiviral therapeutics for a long time. We've contributed to the development of therapies to address hepatitis C virus. And one of the biggest challenges that we faced early on was the fact that there was always this late stage attrition of nucleotide analogs that would go into the clinic, go through phase 1, go through phase 2, go through phase 3, and then late phase 3, when the population boomed, you would actually see 1 or 2 cases of adverse events. Wouldn't it be nice to be able to predict those cases before you actually went into clinical trials? Similarly, and this might be, this example may be getting a little old now, everybody knows about COVID and knows that maybe someone took Paxlovid and reacted in a way, had adverse events because of the ritonavir. You know that the dynamics of your COVID experience was different than your neighbors. And all of this is driven by between individual variation, genetic variation. And wouldn't it be nice to be able to address these kinds of questions in the lab? I mean, it really is the first step towards personalized medicine, especially as it relates for me, development of antiviral therapeutics. So, what is the power of this technique? So this is one example. Polio has a close cousin, Coxsackie virus. Coxsackie virus, it's still fecal oral. This is an enteric virus. It establishes infection in the gut, but then it spreads. Polio only causes disease accidentally when it can access the central nervous system. But Coxsackie will go to the kidney, it'll go to the heart, and replicate. And everything that's been done in animal studies suggests that Coxsackie is a much better virus than polio. But in vitro, in bulk studies, they look identical. Well, when we actually take these to the single cell level with our devices, what we can show is that Coxsackie replicates to higher levels, faster speeds, and the time it takes to get from beginning to end is more constrained, and you can see a statistically significant difference in the start time. It's faster. So by all measures, these data really do suggest that Coxsackie is a better virus than polio, as animal models have told us, but bulk methods have masked. We've also been able to, and this is, again, these are all examples that we pull from the lab for things that we're doing. Our goal is to be able to take patient samples, PBMCs, convert them into induced pluripotent stem cells, differentiate them into cells so that we can actually look at the differences between people. But this is looking at 2 cell lines, Vero cells, you know, those are workhorses for vaccine development, and studying viruses. It's a monkey cell line, interferon incompetent, so interferon alpha signaling incompetent. HAP1 is a human cell line, but it only has a haploid genome. It's kind of like our haploid yeast. We can buy all of the knockouts from Horizon Technologies. But even with this haploid genome, you can see that the monkey cell is demonstrably different, right? Statistically different than the HAP1 cell, the human cell line. So we can see between cell type differences for outcomes of polio infection. We've also been able to use this approach to not only look at efficacy of drugs, but really get some indication of mechanism of action. All antiviral therapeutics, usually start with a screening approach. And you're just asking, does this compound change the amount of virus that's in the cell? Gives you no indication of what the target might be. But using our approach, I can show you that we can distinguish this is polymerase inhibitors from protease inhibitors, from host factor inhibitors. This is a host factor. These are inhibitors targeting HSP90, 2 different ones, ganidaspib and gildanomycin. But you can see they cluster. So, and we've done this enough to be able to say that mechanistically similar classes of drugs will cluster using this kind of principal component analysis of the 2D of the single cell data. This is also useful as medicinal chemists are actually analoging their compounds because you start seeing movement of the clusters when you begin to get off-target effects or you begin targeting secondary targets. Okay, so in this part of the talk, I've told you, we have developed technology to evaluate viral infection dynamics and spread dynamics with single-cell resolution. The population average masked the substantial between-cell variation that exists during viral infection. Traditional approaches also mask infection dynamics, that is the duration of the eclipse phase and the generation time. Stochastic gene expression of the cells influence if and when infection is established. Viral genetics influences the rate and magnitude of viral replication. Phenotypic differences between 2 enteroviruses have been observed, and new insights into antiviral targets and mechanisms have been revealed. Okay, so now to go on to the next part of the talk. What time am I supposed to be finished? 3:45? I'm just, no, seriously, because I do, okay. All right. Okay, because I do have an extra part if I have the time. All right, so we were able to look at, infected single cells. So when Wu joined the lab, he was the one who added the traps. So once we added the traps, the question was, well, can we add more than 1 trap? At this scale, you have remarkable control over fluid dynamics, the flow. So what I'm going to show you here is this is a device with 2 traps in each well. And I can take, this was an experiment where we just dyed cells, a red cell and a green cell, and we could separate the red cells and the green cells. So if we can do that, then that gives us the opportunity to perhaps put an infected cell with an uninfected cell and ask what happens. So I'm going to show you a few movies of the outcomes of some of these infections and share with you some of the directions forward. And all of these are taking us, to think more about cell biology. So the primarily infected cell is going to be red. You're going to see the evolution of green fluorescence. This is an example of a lytic infection. So you're going to see once the cell becomes permeable, lytic doesn't mean the cell blows up, but it becomes penetrable to the small molecules and proteins in the cytoplasm. So you'll see loss of the green fluorescence. And then subsequent to that, you'll see establishment of infection is marked by green fluorescence in this second cell. And this is essentially what we expected for polio. In HeLa cells, it's a lytic infection. And over the decades, as people discover one virus or another, they always wanted to be able to plaque the viruses, right? And to be able to use the plaque assay, you have to be able to, the cell has to lyse. You have to see, you're selecting for either variants or cell types that promote lytic spread. So, that says maybe things are working as we think it might, or should for HeLa cells. But on the order of about 60 to 70% of the outcomes were like this. They were failed lytic infections. So what you're going to see, is that, again, the red cell was stained, the membrane was stained so that we could distinguish it from the infected cell from the uninfected cell. This cell is going to turn green, it's going to lyse, but the second cell never becomes infected. And so for us, we were, puzzled at first, but then we're like, this clearly, when you look at the kinetics, the start time was later than those that actually did spread. And so the thought was, well, maybe what we're seeing is activation of intrinsic defenses in the cell, innate immunity, and release of interferon. That's protecting the secondary cell. And most of what we know about pattern recognition receptors and all those signal transduction cascades that lead to an antiviral state for a cell have been gleaned from studies of using knockout mice. You cannot see these differences in cell culture until now, I believe. And so this is a major shift for the lab, and one of the reasons that we moved to UNC. Because of the strong innate immunity community. So HAP1s are interferon proficient, Vero cells are not, and you can see that if we knock out the interferon alpha signaling, the number of infections that fail to spread go down relative to interferon proficiency, the 60% here. Does that show? No, this thing's in here. So we're comparing 60 to 35 here. Okay, now this last experiment that I'm going to show you really blew my mind. You're going to see that infection is established in the primarily infected cell. But before there's any lysis observed during this primary infection, you're going to see the uninfected cell turn green, suggesting that we're looking at non-lytic spread of the virus. And Carla Kierkegaard at Stanford began talking about, non-lytic spread, I don't know, well before we made this observation. And I give her credit for founding this field. One of her trainees, Bill Jackson, who's at Maryland now, has continued. Nihal Alton-Benay, who is at NIH, is also a major contributor to this. What we know now is that polio, other enteroviruses, transmit through vesicles. So we're bypassing this lytic mechanism, and viruses are being taken up in an LC3-dependent manner into autophagosomes. Those autophagosomes, maybe I should use this. Those autophagosomes can fuse with endosomes to create amphisomes, multifascicular type bodies. And then these things can fuse to release unilamellar vesicles phosphatidylserine coated that likely use some type of phosphatidylserine receptor for attachment, but uncoating nevertheless still requires poliovirus receptor. So when we knock out the poliovirus receptor, we still don't see any of the spread. So this is what I mean by, I'm not sure if I have the wherewithal to get into the fray here, because there's no consensus in the autophagy field about how any of this stuff is happening. And now I'm adding a virus on top of it. So with that, this is my conclusions. Essentially what I'm saying is that if you can do an experiment in a cell in bulk, adding a single cell dimension where you're monitoring dynamics of whatever the process is going to be illuminating. I've shown you what we're doing, but we really want to now use this to understand better the mechanisms, of virus transmission. We're interested in learning a lot more about innate immunity and distinct antiviral therapeutics. And of course, none of this would be possible without the people in the laboratory. And this work really was started at Penn State before I left, and continues today with the people in this lab, and have all of the modeling has been done in collaboration with Klaus Wilke. So with that, I think I have hit 40 minutes or thereabouts. I might have time for questions, but thank you for your attention. It's really a pleasure and privilege to be back.
Questions. It was illuminating. Thank you.
Fascinating talk. This question is based on my own lack of understanding of single cell stuff. So you showed us that the population behavior can be decomposed into individual single cell behaviors.
Yes.
So is there a representative kind of phenotype that you'd say in whatever biology process that you observe, or in the single cell field, do you consider all of these individual behaviors as somewhat more representative? In other words, is there one phenotype that you focus on?
Yeah, so for us, so the question really is whether or not there's one, phenotype that we pay attention to when analyzing our single cell data. It is the start time, right? Trying to understand virus-host interactions, restriction factors, they're going to perturb start. You don't see that in bulk. And so it has really given us a bird's eye view of genes, both viral genes and host genes that are required to make start as efficient as possible. And that really has not been feasible using cell culture methods. Yeah.
I think that was really cool. I love the system. My question is with regard to the cell biology. It seems like your fluidic system is an ideal single cell mechanism to set up some type of CRISPR screen to identify potential co-receptors for that non-lytic entry. I'm just curious as to your thoughts on that.
Yeah, so we have not had to do a screen at this point. We have evidence that TAM receptors are involved. So TAM receptors, Shelley Earp, you probably know from UNC. He's been looking at one specific one over the years. So there are a lot of small molecules that stop signal transduction, through TAM receptors, and most of those prevent spread. So that's very in the early days, but Crystal Starbird is the new structural biologist in our biochemistry department who's working on that with us. She's taking the lead on that. Yeah.
Hey, Greg, really nice talk. So I wonder, the lytic versus non-lytic effect, can that be caused by the the different variability of your virus when you're infecting a single cell level. So you see, some may be more toxic versus not others.
Yeah, so we really believe that lytic infection is just the consequence of the time, the duration, right, of the infection. Most things, the non-lytic phase, I didn't have time to show you a lot of the dynamics. When we stop, and we have ways of stopping non-lytic spread is the fastest mechanism for spread, and it's a much higher multiplicity. When you lyse, the virus, free viruses go everywhere. We think it's off path. It's just the way we've set up the experiment. If you go 24 hours and let anything express that long, the cell dies. And so we don't really think it's a relevant mechanism.
If I've exhausted hands, I want to thank you again. I'm going to come around here and let you receive this. So you
Wasn't expecting anything here.
remember this heavy gift from.
Hopefully you guys will mail it to me. Won't make me put it in my.
Class of 93 Dean's Distinguished Lecturer, October 9th today. So
Thank you.
I have one more bit of news for this afternoon, if you'll allow me, and that is that, thank you, that at this time of year, as dean, I have the opportunity to do one special item, and that is to honor an existing faculty member with the Case Medal for Excellence in Health Science Innovation. This is what we like to describe as the highest award given by the School of Medicine, and recognize a member of our faculty who's had extraordinary impact here and on the field of medicine. And past recipients of this award are substantial individuals. I'll mention a number. Mark Griswold, Hunter Peckham, Jackson Wright, Fabio Cominelli, Pam Davis, Cliff Harding, Maria Hatzoglu, George Dubiak, Paul Tizar, and last year, Hope Barkukas. And I am delighted, I hope to surprise this year's recipient, you never know, as Dr. Alan Deal. I'm going to have it is designed as a surprise, and usually I pull it off as such. So I've got a few minutes of conversation about Alan and why this is actually so special for both the department and for the school, and that'll allow Alan to think about what he really wants to say and then forget that and say a few other things. So, of note, I listed a number of names to you. Only two prior individuals have actually had the title of and the responsibilities of department chair, and I want to explain why that's especially appropriate for both this department and for Dr. Deal. Dr. Deal came in 2019 as the Leonard and Jean Keggs endowed professor and chair of the department after a national search. And the following year, he became the deputy director of the Comprehensive Cancer Center and was essential in helping that cancer center get its renewal application, and has helped focus the Department of Biochemistry in the area of cancer biology. I want to comment about the Department of Biochemistry that most of you in the room know so well. It's a pretty amazing history of a department, and he set out to both consolidate and then rebuild the department, who has had innumerable figures over the course of the past 90 years. And I'll mention a few for the biochemists in the room. Harlan Wood, who's credited with defining the role of how carbon dioxide in animals and human metabolism is important, something called CO2 fixation. And most critically, the conversion of pyruvate to form oxaloacetate, important in the TCA cycle and in biosynthesis pathways. And then Mert Utter, who was the chair from 1965 to 1976, worked with Harlan Wood on pyruvate carboxykinase and carboxylate. And then Richard Hansen, after an interim, became chair in 1982 through about 2002. And many of us are honored by both his presence, serving under him, as Craig did during their graduate program. Richard was a remarkable individual and would give our medical students and our PhD students hours-long lectures in intermediary metabolism. Whenever he came up to the podium, he took off his jacket because he would splatter the chalkboard with equations and with formulas, and he would get his shirt, let alone his jacket, filthy with chalk as he gave his talks. And how he remembered all those synthetic pathways without ever getting it wrong is beyond me. His own research was in phosphoenolpyruvate carboxykinase, an enzyme which is key to gluconeogenesis. And I remember that he created a transgenic mouse that overexpressed that enzyme in the muscle. And for those of you here during that time, that mouse would run on a cage for hours and hours and hours and never get tired. So he really showed that all it took was a little glucose. I also would point out that some of his distinguished graduate students were Jay Short, who now is active on the West Coast with his 7th or 8th different startup company, Tony Winchot-Boris, and Maria Hatzoglu, who I mentioned as a prior recipient. And by the way, Shirley Tillman, who served as president at Princeton. So how in the world could Alan take the job after those distinguished individuals and lead a department? So Matt Allen received his BS summa cum laude and then PhD in biochemistry from UNC State and then the University of Missouri, Columbia. Was a fellow with Chuck Shear at St. Jude's. And any of you who might remember Chuck Shear, also a incredibly insightful, hyperactive, chain-smoking tumor biologist. Up with that guy. But Alan somehow did. If I'm wrong on that, let me know. That phenotype actually doesn't exist anymore. But Chuck was a motor force. His own research, Alan's, is in cancer initiation and progression. His lab explores the role of lipid signaling, epigenetic regulation, CDK, cyclin D, CDK4/6, E3 ubiquitin ligases, and its role in tumor suppression. The implications of that work are really quite profound, and a number of clinical studies have been brought forward with those targets. I would be remiss if I didn't remind you just how important his service has been for our cancer center. He also serves on the external advisory boards for 5 other NCI designated and aspiring cancer centers. He's been continually funded without interruption by the NIH. Has more than 160 peer-reviewed papers, 40 review papers, and has had more than 2 decades of funding together on 2 different program projects. So in addition to his own work as a walking the walk, superb cancer biologist and biochemist, he's done a remarkable job recruiting and rebuilding the Department of Biochemistry. And that's, I think, what I really want to highlight. First, he consolidated the more independent Center for RNA Science and Therapeutics into the department successfully as it was lingering and not sure where it wanted to go. And he's recruited incredibly well. Ryan Arvidsson, Kai Cow, Leah Gates, Nathan Howell, Chris Hubert, Tai Kim, Chai Lee, Shin Lu, Joe Luna, Boaz Tairaj, Yi Zhang, and Wenjian Gan. A total of 12 faculty over the past 6 years, more than any other department. Don't worry, he'll be asking for more after this award. And as a result, the Blue Ridge ranking, which frankly, I'm sure with this group will do even better, has risen under his leadership from 44 to 16 amongst departments of biochemistry nationwide. In addition, when he got here, he said, so tell me about this undergraduate program. What's that? And that program has flourished under the department's leadership. There's now an honors program. Its growth has been about 30%, and the number of students, many of us at the faculty level have biochemistry students in our laboratories for the summer. It's been an amazingly successful and trajectory department for many undergraduate students here at the university. So, Alan, it might even be said that this is a little late in coming, and I'm going to have you come up and receive this small medal. This is J. Allen Deal, PhD, 2025. And you'll notice it's a little heavy because it actually puts some money on it. Don't melt it down too quickly. And if you want to say a few words, we'd be delighted. So thank you.
Thanks so much.
Let's get a picture.
Let's get a picture. Thank you so much.
Wow. Don't really know what to say other than I'll thank Stan because as I reminded him earlier, he was actually the chair of the search committee when I got here. I don't know if he wants to actually be reminded of that. And I'm going to do something that I tell people to never do when you're introduced by someone. I'm only going to correct you on one thing. We're actually number 12. But I'm sorry for doing that. But I think one of the reasons I comment on that is because I think what I'm most proud of is the development of the department. Huge. There's no way I can fill the shoes of my predecessors. I don't even try. That's not my role. I came here to do my best, and I'm incredibly proud of the department, the school, and the university. And I'm very grateful to Dr. Gerson for giving me this opportunity. I think you can't do this. No individual is capable of doing this. When I arrived, the faculty that were here, that knew Dr. Hansen, embraced me and the ideas that we had. And as a consequence, I think the faculty that have joined, chosen to take a risk with a long-haired chair. I'm really grateful to. They've done an amazing job. The Blue Ridge ranking reflects their activity because it's a reflection on NIH funding. It's not my funding; it's theirs, and it's the science that they produce. I also want to point out the activity and the efforts of the staff that we have in biochemistry. They do all the work. You know, the chair sits back and says, I have this idea, I have that idea. And they have the responsibility of going out and getting everything done, and the faculty have to do the work. So this while I have this really belongs to the entire department, and I'm incredibly grateful for that. I do want to, I see now some friends. Very grateful to my wife and one of my mentors, Dr. Serge Fuchs, and his lovely wife, Lena Fuchs. Raise your hands, please. Have been with me from the beginning and I'm very grateful to them. And rather than babble on anymore, thank you very much. Congratulations.
We have a bit of a reception outside, and then I'll remind you that we do have the graduate student presentations a little later this afternoon, so please join us for that as well. So thank you and congratulations.
See you.